I showed you the major types, okay? The silent, the nonsense, missense, and the frame shift mutations. Now what else can cause, except for the mistakes during the replication, what else can lead to the mutagenesis? First one is so-called intercalating agents, like this molecule, the acridin. Huh? That's good. That's awesome. I may ask you to turn it off in maybe 5 minutes. Okay. Now, intercalating agents. Remember, when we talked about the structure of the DNA, we noticed that pairs of nitrogenous bases, okay, they form kind of a ladder. They stack one on top of another. So what happens, intercalating agents insert some molecules of intercalating agents. They get between the nitrogenous bases, and when DNA is replicated, that can cause the frame shift mutations, because when DNA polymerase cannot read the two adjacent nitrogenous bases, okay, separated by the intercalating agent, it will skip it. There's going to be mutation, and that may cause a problem. Okay, a couple of the common intercalating agents are ethidium bromide and acridin orange. So they are extremely carcinogenic. Ethidium bromide is added to visualize DNA molecules in the agarose gels, and it must be handled exclusively in the gloves. There were cases when people handled it for several times without gloves, and they got skin cancer, and they had to, get treated. It's really, really not a pleasant thing. Acridin orange is the stain that is sometimes used to visualize structures in the mammalian cell, especially DNA. You can see orange dots of DNA under the microscope. Really helps. It's not going to explode. Don't worry. Don't worry. But those two, if you could turn them off, yeah, these two, it would be lovely, because that's it. That's all we need. Okay. So acridin orange is used in the lab. Again, you can work with those agents, but you should be cautious and don't touch it with a naked skin. Another, now you can see that actually exemplifies when there's an insertion, these nucleotides are being missed, so you have a frame shift mutation. Another common mutagen is the radiation. There are two major types. So ionizing and non-ionizing radiation. Ionizing radiation may cause strand breaks. And if single strand break is fairly easy to fix, as you can imagine, right? Because this break is going to be detected by the system of repair enzymes that will cut the piece of the DNA out and replace it based on the unchanged complementary strand. Double strand break is much harder to fix. It can be done, though. It can be done based on other unaffected DNA molecules. Does that make sense? Without getting into too much molecular details of what's going on, imagine this. If there is a fragment of DNA that has double strand break, repair system sort of copies that fragment from another DNA molecule, completely different double-stranded DNA. Does that make sense? Like if you have breaks in 2 strands, then another DNA molecule, another double strand is used to proofread and replace. It goes through the recombination. So the idea is to copy the part of another DNA molecule and insert it instead of the broken one. Now, one thing that you have to appreciate regarding all of these repairs, and we'll talk about thymidine dimers in a second, is that when you go and get the lung X-ray or chest X-ray, are you going to get some double-strand DNA breaks? Oh yeah, oh, be my guest, sure enough. But you have a few, and your repair systems have a lot of time to deal with that. Okay? When you take a flight, you are exposed to radiation, trust me. You get few double-strand breaks, but you're going to be fine because your repair systems have enough time to deal with that. But if you constantly are exposed to a certain mutagen, whether it's radiation, whether it's something else, okay, you increase chances for those mutations to get fixed in the genome, to not being repaired. Does that make sense? Okay. Now, UV light. That's the mutagen that we probably are exposed most to. We go outside, you know, we play in the sun, and we catch some UV light. It causes so-called pyrimidine dimers. Usually, it links to thymine nitrogenous bonds, nitrogenous bases. Now, you can imagine that these dimers, like this, they are not going to be replicated, they're not going to be read by DNA polymerase III properly, and that will lead to the insertion of the mutant residues, maybe the frame shift or something like that. Does that make sense? Could you please turn off that, and we're good. Yeah, this one too. They're good. Okay? Does that make sense? So you have 3 major ways to introduce mutations. One is intercalating agents that insert themselves between the nitrogenous bases. Second is non-ionizing radiation. Third is ionizing radiation. And they all have different mechanisms, right? So please have an idea about the mechanism. Skipping the nucleotides, different types of frame shifts for intercalating mutations. Double strand break, which is pretty brutal, for ionizing radiation. Or thymidine dimers, pyrimidine dimers for the UV light. Yes? I am not entirely familiar with the technique that they use. What I know is that you can manipulate the genome by there's a process called recombination, and okay, let me try. Okay, no, it's just, again, you have to imagine things. So what is recombination? So you have double-strand DNA. Okay, imagine you have a replication machine here, and it goes, and I'm not going to picture all those forks and stuff. So imagine it moves, say, this way, and then close to it, you add another molecule of DNA, which here and here, okay, it's exactly like this part, for instance. So a DNA polymerase may accidentally jump over here. just by chance. Okay? Because it's the same sequence. And then it will copy this fragment, which has nothing to do with an actual genome. It will continue because it doesn't know any better. Does that make sense? And then it will jump back. This is, to extent, a random process, the recombination. But scientists have certain tools to make sure that this recombination, kind of sneaking in the gene, happens in the very exact place of the genome of the plant or of the animal. Does that make sense? So they specifically introduce the gene that they want in a very specific part of the plant genome, and then they manipulate. I'm not a great plant biologist, so they manipulate whatever they manipulate in plants, seeds or whatnot, okay, to introduce that altered genome into whatever. Like in mice, it's going to be, they will introduce the altered DNA into the oocyte instead of the original genome. Okay, they will kind of substitute. Does that make sense? So then when they do the fertilization, you have heterozygous mice, but when you have many heterozygous mice and you start to breed them, eventually going to make homozygous mice that will lack the gene or have the gene or whatnot. So it's fairly complicated but fairly routine technique. But double strand breaks, I don't really know if they employ double strand break mechanism. Not really familiar with that. Now, those are some the exemplary mutagens. So analogs of nucleosides. Okay. So they convert one base pair to another. Okay, they are inserted. For example, 2-aminopurine is inserted instead of adenine, but then causes the pairs with C, so it switches AT to GC. Okay, same goes for 5-bromouracil. Okay, modifying agents. Nitrous oxide converts GC to AT. Okay. Intercalating agents. That's what we just discussed. When they insert, those molecules are inserted between adjacent pairs of nitrogenous bases, they cause frame shift mutations. Ionizing radiation. So two things. First, single and double strand DNA breaks. Again, when there is a repair, it may not be perfect. May or may not be. That's what we just discussed. Okay, the double strand or single strand breaks. They also can modify the bases. And ultraviolet radiation, thymine dimers, okay, frame shift or point. Now, here's the take-home message. What you have to know, you have to be able to explain what's going on during the ionizing radiation-induced mutations. What type of mutations UV light causes, and what intercalating agents do. But I will not ask you like the structure, okay? Be able to describe what's going on, like GC to AT mutation. Is that a frame shift or a point mutation? Point. So you have to be able to tell me that, okay? Deletion of a nucleotide, or deletion of a pair. Is that a frame shift or a point? It's a frame shift. Does that make sense? Okay, what type of, like, ultraviolet, what does it cause? Formation of thymine dimers. It's going to be probably frame shift, but can be both actually. Double strand break can be all types of mutations. So I will not ask you to distinguish those, okay? So repair. Let me see what we've got there. You know what? We're going to actually take a pause here.